Thanks, Martin. Thanks in the first place for inviting me to this seminar. I guess almost all of us are Polish speakers, so Martin, of course, asked me to talk in Polish, but since I'm born close to Katowice, I told him I could do Silesian, but Polish I'm not sure about. So I would prefer to give the talk in English, also because all the research, of course, was done in English. And as you can read from the title, it's an attempt of me and my supervisor at IST Austria, to derive a very well-known and prominent, well-known I should put in brackets, a very prominent and important genealogy system that is active in the early development of the fruit fly embryo from first principles, from physical principles, by optimization, by an optimization rationale that I will explain in the following. And I should mention that this is in collaboration with William Bialek at Princeton University and Thomas Gregor, who used to be in Princeton and now moves his group to Institut Pasteur in Paris, and Alexandra Walczak, who is in Paris now, and Mariela Petkova, who is a grad student at Harvard mainly, but also at Princeton. So I would like to take you directly into the topic. I hope that you can see the video playing here. So what you can see here is the early fruit fly embryo, about 1 to 2 hours after fertilization. And as every embryo, it starts out from a single cell, so it's an egg, right? And in the beginning, in this big bag, which is called the syncytium, which can be considered one cell at fertilization, there is only one nucleus, and it keeps on dividing about 10 times. And that's the process that you can see here. I can replay this. But the point here, the main point is that up to these 10 divisions, which is about 2 hours after fertilization, you can take out each of these nuclei, and you can put it somewhere else, and it will not affect the correct development of the embryo, which means that all of these nuclei are not specified, not differentiated yet. So when you look at the state about 2 hours later, you can see very distinct positional cues that have formed. The back of the embryo developed differently than the front, and we also have some distinct development of cells in the perpendicular direction, and that means that somehow during the course of these divisions, or at least at the time point afterwards, these nuclei must have acquired some positional information. They somehow suddenly know where they are, and they must have read out some signal that told them, you're so far away from the anterior source or anterior pole, you will become part of the trunk, for example, of the fly, and you will become part of the head, right? And it is well known in developmental biology how this can happen. So people have studied, particularly in this system, the processes that specify the different development of the nuclei very well. Since the 1980s, there was a Nobel Prize given for that at some point. And the process is the following. In parallel to these divisions that I showed you, the mother fly puts some mRNA of a particular protein, a morphogen protein called decoy, at the anterior pole of this embryo. So I had this thing, let me take it, here, right? And then directly after fertilization, this mRNA starts to be translated by ribosomes, which are all over in this embryo. So we have a source of production here of proteins, and these proteins can diffuse through this syncytium, through the embryo, and they are also degraded. And then you can do some simple differential equation-based math that will tell you that this results, at least in stationary state, in an exponential gradient like this. So the mother fly makes sure that in parallel to this division process, an exponential gradient forms along this anterior-posterior axis of the embryo, and of course this concentration signal can be read as a distance measurement signal, right, by genes that are activated downstream of it. And there is a particular well-studied, but still not well understood layer of genes called the gap genes, which is activated downstream of this morphogen gradient, these 4 genes, and they start forming localized patterns, which then define some kind of positional code along this axis, in the sense that nuclei that are residing at different places along this axis can read out different combinations of concentrations of these genes, and this way they can determine how far away they are from the anterior pole, and they can determine their specific fate and what to become in the further course of development.
Head on the right.
Sorry?
The head is on the right. The head is on the right.
The head is, I think the head is actually on the left, but I'm not sure about this because I'm not interested about this. I'm just interested about this code. Right. Now, a couple of years ago, there was some very important work, I would say, coming from the lab of Thomas Gregor, where they managed to stain these 4 genes simultaneously by antibody staining. And this way they could determine in individual embryos, of which they measured many, the variation in these concentration signals of these genes, of these gap genes that are activated downstream. Right? So here you can see the measurements for the individual genes. And you can see that there is some spread, right? And I should highlight that these bold lines are not the averages, but that is random picks from the ensemble of measured patterns that were obtained by this simultaneous staining. And you can see that in general, except for certain parts like here, the code is very precise, right? Having these measurements, of course, you can calculate the error bars here, right? Standard deviations, and they have done this. And you can do all kinds of calculations with this. But when we say this code is precise, as physicists, we would like to ask, is there any way of formalizing this, of quantifying this, let's say, with one number, right? Such that we could compare different types of these codes, maybe also in other species, and we could easily compare their precision by some measure that we will define on some scale, right? And there is a way. They have formalized this, and in order to explain how this works, I would like to just motivate this by a simple cartoon. So here we have these 4 genes again with the error bars, right? And now imagine you are a nucleus located at 30% distance of this anterior pole. Then, of course, you will read these concentrations, 0.6 of the yellow gene and 0.9 of this red gene, right? And if you are a nucleus further towards the posterior of the embryo at 60%, you will read other concentrations of preferably other genes, right? So this way, of course, you can determine your position, your distance from the anterior source. However, the whole point, of course, is that these concentrations are not well-defined numbers, but they are fluctuating, right? Between the embryos, right? And even when you try to maintain a constant concentration at some point in one embryo, you will have a limited number of proteins, which means that the readout also will be noisy because the random arrival of these proteins to a downstream promoter that carries on that information also will depend on stochastic processes. So we have some error bars here, and these error bars translate to error bars in the estimated or inferred positions that the nucleus can read. Now, if you take that perspective, you can use information theory, but in particular, you can define something that we call positional information. You can formalize positional information using a standard measure from information theory, which is called mutual information, given that you know the joint distribution between these inferred positions and the concentrations of these 4 genes, which then you can imagine are something like a multivariate Gaussian distribution. So when you have this distribution, you can integrate over it like this, and you have to assume, so you also can calculate this, of course, when you assume a prior distribution for the inferred position. And the best you can assume here, and of course that is also what the nuclei are facing, is that this is a uniform distribution. So when you do this, you can, from this data, compute this joint distribution, integrate over it, and you get a number in bits when this is the log 2. You can also take a different log here, then it's not bits, but when you take log 2, it's bits. And it turns out that this pattern then encodes about 4.3 bits. And if this number doesn't tell you anything, so I'm putting this other measure also there because I'm often talking to biologists, they don't know what 4.3 bits would mean, then you can convert this number, or actually, I should say you can also from this joint distribution, you can compute also something like a positional error, which tells you what the estimation error in this inferred position is, and that turns out to be about 1%. More concretely, they have done this in that study for all the positions along the anterior posterior axis, okay, the inner 80% because things are not well measured at the poles. And when you look at this positional error, and here you can see it for different combinations of genes, when you take only one, then it's very high. In some places here, it's actually still very low, and you can increase the number, but the really interesting thing is when you take all 4 of them, then almost everywhere along the anterior posterior axis, you reach a precision of about 1%. And that is well below the internuclear distance, which is something like 1.4%, because we have about 80 nuclei along this axis. And that suggests, together with the fact that this is almost a uniform distribution, which is questioned by more recent measurements, I should say, but let's assume for the sake of this talk that we can observe this, that suggests that somehow this system has been optimized to convey the right amount of information that each of the nuclei need to specify their position with not with 0, but with a reasonably low error, such that each of the cells on average will know what to become in the later course of development.
But the imply is not one-dimensional object, but three-dimensional. So
yes.
It's missing the dimensions. Also, abdomen definited sometimes gradients.
There is another patterning system which is orthogonal, right? That takes care of that.
Also,
but
the distance in that right, but some symmetry, but defining distance from the axis. So,
I'm not sure I get the question. So for the 3D patterning, you have other orthogonal patterning systems, which partly interact with this, but I'm not sure how much they would affect actually the. So we believe, since you can show this using these 4 genes, right? You get this error. We believe that you don't need more to actually specify the axis in one dimension. There may be interactions in the other dimensions, but they may be. We don't believe they're essential, right, for achieving this precision.
It's about thinking about flights. It's not just
no.
Yes, it's more complicated.
It's more complicated, but there's also other processes coming on top. For example, there is, after this patterning has happened, there is another layer of genes, yet more refinement. This information is passed on further downstream, and errors are even later corrected by mechanical rearrangements and so on, right? But this is the first step, and we're interested specifically in this. It's not all the story about the fly, obviously. Now, since we have these signatures of optimality of conveying information, positional information in an optimal way, we may ask, can we actually derive this observed gap gene pattern and the network that generates it just by optimizing what we believe to be the main objective of this system, which is positional information, right? Can we predict this starting from biophysical principles by just maximizing positional information in a biophysically motivated model of this patterning system? And if yes, if we can do this, the second question is this a unique solution that nature has found, or is there many solutions to that problem, right? So that's the main objective. That's our main working task that we started to follow up on. And the way we're going to do this is we're going to consider this gene regulatory network as a black box that somehow we have to construct from biophysical principles. And of course, we will use the same information measure to define positional information, and also in order to become able to compare to the experiments. And then this whole network, the whole embryo in a sense at that stage of development, will become a noisy info transmitter where the position is encoded first in this exponential input gradient. Then there's 2 sources of noise that hamper transmission of information through that gene regulatory network, and that is input noise, as I mentioned already initially, that results from finite concentrations and random arrival of proteins to their target promoter. That already creates some type of noise, which we call input noise. And then, of course, there is stochastic processes that generate the output patterns, which is protein, well, first of all, mRNA production, then protein production, and so on. Diffusion, right? That puts on top some production noise, which we call output noise. And that creates the error bar around these gene patterns, right? Or expression patterns. Now, then the task, of course, becomes, when we have taken the setting, to create this black box, right? So for that, in order for the whole approach to work, in order to be able to derive this from physical principles, you of course need a model that is based on physical principles. And we basically, so most of the things that we have been doing in the last years boil down to creating that model and refining it more and more. So ultimately we ended up with a model which is necessarily spatial, stochastic, and dynamical, because that are the features of this developmental system. Secondly, the main features of the model are that the output are the gap gene profiles, the expression profiles of the gap genes, plus the noise bars, importantly, the noise bars around them, because only when you have also the noise bars and the average profiles, you can compute this positional information from the joint distribution that I mentioned before, right? And most importantly, we claim that our model is the first of this system that is biophysically accurate, and it has about 50 biophysical parameters which all have a biophysical meaning, something like a binding energy, diffusion constant, degradation rate, and so on. And it features all of the things that play a role in creating these patterns, like gene expression, stochastic gene expression, I should say, stochastic degradation. It also, in a rudimentary way at least, takes care of these nuclear divisions that I have mentioned initially. And there is regulation by morphogens, which is necessary to create these broken patterns. Otherwise, you could only create patterns that stepwise come down at certain concentrations of the input gradient. It has diffusion in there, so it's a spatial model, right? And it has, and that's important, it has biophysical models for all the noise sources that create the noise bars around the average patterns. So having this model, then you could do two different things, right? The classical thing would be you just take the data, which we have from the lab of Thomas Gregor, right? And you could just fit this model using the measured gap gene patterns, and then you could infer one or many gap gene networks, sets of parameters, that match the data well. However, that's not what you want to do. We also will do this at some point partly, but what we really would like to do is to optimize this model in a way that we change these biophysical parameters and then monitor this positional information objective function and filter out the solutions that maximize it, right? Maybe they do that in a unique way, we don't know. Most likely there will be many solutions when there's so many parameters. There's a very rich phase space that we can explore. But this way, we get predictions for optimal gap gene networks that maximize the encoding of positional information for certain sets of biophysical parameters. So that's the task. Now, in order to show you how this works in a more technical way, I would first like to talk about, just briefly, about a simple model that we created, which implements this idea already completely, I would say, only that it looks at the reduced system where we have only one input, this input gradient, and one output gene, G, which is just a step-like pattern because other patterns you can't create with only one output gene. And so we implemented this on a spatial lattice, which is cylindrical, which is an approximation of the geometry of the embryo. In each of these little volumes, we simulate the random arrival, or we actually don't simulate, but we model the random arrival of the input proteins to the promoter of this one regulated gene G. Then we model the random production of this gene, the random degradation, and most importantly also the spatial coupling between neighboring volumes via some hopping rate based on the diffusion constant of the protein. And the way we model it is via a set of coupled stochastic differential equations, Langevin equations. So here's the ansatz, right? So we can of course indicate all the volumes of this spatial lattice by some index I. And then we have in each volume a production function for the population of that gene G in volume I, which depends on the input concentration at that position, and it can in principle also depend on its own concentration because of self-regulation, which is possible in biology, right? Then we have a coupling term, so that's the spatial coupling between neighboring nuclei. So this N index here goes over all the neighbors of volume I, And this is this hopping rate that I mentioned, which is simply the, I can take away all of the noise essentially, right? And then any profile will encode, if it's not flat, it would encode information perfectly along this axis. So everything that we will be saying about this will be for specific choices of these constraint parameters. Some of them we know, or at least we have a good guess for them, like for the lifetimes, and we actually have measurements for the input gradient, but we don't know what the output protein number is. So that is something that we have to impose. And the way we did this is we have chosen reasonable parameters that are in line with the measurements, that give error bars that are very similar to the measurements. But that creates a setting that determines, let's say, some maximal value of information that we cannot surpass. It does not determine yet the shapes and the regulatory network, and that's the thing that we want to get by optimization. So, and I should mention, we can go from here to here in order of seconds, which we consider some kind of technical achievement by itself, because before people were running long simulations of that system to get the covariances and so on, which could take up to weeks, right? So that's just mentioning beside. Now what can we do with this then? So what I show here, just this one slide, is still for this simplified model, where I vary these regulatory parameters of the input-output relationship, so the steepness and the activation threshold here, and on top, in the different panels, the diffusive coupling is increased. Right? So first of all, we can see that in each of the panels there is a well-defined minimum, right, for these two parameters, where information is maximized. And then we also see that as diffusion is increased, we get still more information, and the shape of this phase space changes. But when we increase further, when we make the patterns too flat, then again the information is decreased. It smears out here. That's because this is the steepness, so at some point it doesn't matter whether I make it infinitely steep, I just smear it out by diffusion all the time. And of course when I make diffusion super strong, at some point I get a flat pattern in that system, and that again encodes no information, right? So there is not only optimal parameters for regulation here, but also some optimal spatial coupling. Now I told you that this is not the end of the story, because all of these genes, first of all we have 4 genes and they are interacting with each other. And if we want to extend the model to this, of course we are facing the problem that the number of parameters increases with the number of genes squared, because all of them in principle can interact, right? But we did this, and in view of time, I'd like to go directly to the full model and show you how you can extend the simple model. So it's essentially just a bit of bookkeeping. So you make a couple of extensions. You just add some index alpha here, which is the gene species. So you will have 4 of these now, right? For each volume, 4 species of genes or proteins in each volume. Then also we assume that there is more inputs than 1 potentially, because that also is something that is well known by now. And that's all of it, so we will just get more equations. And we assume a more realistic regulatory function, which is also essentially a sigmoidal function, but it has this, this energy term here, which is more thoroughly rooted in statistical physics. And we do that because if you would use these Hill functions, we would have to impose, is it some multiplicative regulation? Let's say when we have 2 inputs, do the 2 functions multiply, do they add up? In that setting you don't have this problem, you just add up binding energies essentially, and there's some baseline energy that you have to specify. And for any regulation you have a well-defined way how they combine in creating this input-output relationship. That's called the MWC model. Now, there's a couple of other things that on the way we had to take care of. For example, we figured out that with the setting that we had, we never could get patterns that go down here and here. So we had to actually include 3 input gradients, which is something that is known from measurements. So we found this to be necessary to be able to create patterns of that kind. And also at some point, because we found that we had not enough noise for these constraint parameters that we chose, we had to include another type of noise, which comes from bursty production of proteins from their corresponding mRNA population. And the way we do this is we make another assumption. So essentially, the equation that I showed you before now models the mRNA population, and to get the proteins, we make some, let's say, adiabatic assumption, in the sense that the mRNA is longer lived than the protein, such that we essentially the protein population tracks the mRNA population in that limit, right? And then we can just add a beta factor here. So beta is the burst size, and the number of proteins produced on average from one mRNA. And then you have to take care of a couple of details, but you can work out the equations also in that setting. Now, here I show you, hopefully, yeah, this video is running, the output that we get from the model, right? So it's a dynamical model. We start with this, for a certain set of parameters, we start from 0 concentration at time 0. Now these concentrations ramp up. So this is modeled now, right? I will tell you in a second how we obtain the parameters for that. And then we stop at some point, right? We get these mean profiles, but most importantly, for any such run, we can also get the error bars, right? And of course, I chose a set of parameters that we obtained that comes very close to the measured one, right? So here you have the comparison, right? So the only thing that we are missing here is this very posterior bump, which we cannot create. I can comment on this later if you ask me. But now I will show you how we obtained these parameters, which was actually done by optimization. So with this fully extended model, we have about 80 model parameters in total. Some we know roughly, some are set to reasonable values, such as lifetimes. Still, we have about 60 parameters which have to be optimized, right? So we cannot just do this by screening, of course. We do random optimization. For now, it's, and it probably always will be, simulated annealing. And we start from random initial conditions, but to our own surprise, this worked remarkably well. And I will now show you the optimization trajectory that has produced this set of parameters that I just showed you in the model example. So starting from random parameters here, we get, for example, something like this, where all genes are down, but one is activated everywhere. Of course, this pattern carries 0 bits, no position information, right? Now, as optimization progresses, so here you can see the progress of optimization steps versus the objective function, which is position information in bits, right? Very quickly, we start finding these broken patterns here, right? And of course, they will be filtered out because they constitute a substantial increase in information. And you can see as we go further, we start literally adding in more and more stripes into this, right? So for example, here we broke down also this one, and then the yellow guy was activated somehow, but you can already see that this has to do with the interaction between the green and the yellow guy. You can't create these broken patterns just by coupling to this maternal inputs, right? Same for the third pattern that's added in. And finally, we arrive at this optimized pattern, which happens to carry 4.2 bits. And you can see that it is indeed very similar to the measured pattern. So the question about can we derive this pattern somehow by optimization, we claim to be yes, and we can make further comparisons, right? For example, we can look at the noise bars. You can see that also these turn out to be very similar between optimization and the measured patterns. Then we can even look at this one measure that I had initially, which is the error bar, the positional error, sorry. And you can see the more recent data actually doesn't show that it's uniformly 1% everywhere. It goes up even to 5% in some places. We have these peaks. But yet our pattern, the optimized one, actually captures this quite well, right? And most importantly, in most of the parts, it also brings down this positional error to 1%, about 1%. We can also look at the decoding maps, which is the implied or inferred position that the nucleus can read from this noisy pattern versus the actual position where it's located. And also you can see some good correspondence between the data and the optimized pattern here. Now, as I said, we claim that we can derive this, but is it a unique solution? Right? That was the other question. And there it turns out, no, it's not. So we get a lot of other patterns that look somehow similar, but there's actually a large variety. And just plotting here a small choice of them, I can produce hundreds of them, right? And they will have some common features, but in particular, the order of the stripes will be different. Not all of them will correspond to such patterns. And now you may wonder, okay, how did I ever find this one, right? And I will tell you how I did this. So we have a little Lagrange multiplier that we add to our objective function, such that the distance between the measured pattern and the optimized pattern is also reduced upon optimizing position information. That Lagrange multiplier is small, right? So we are still optimizing position information, but we can use it to filter out solutions that look similar to this one. And even without, you can get patterns that recover some of these features, like this one, for example, right? So when you just exchange the order of stripes here, for example, if you would move the blue one a little bit to the posterior, and the yellow one would not be here, but where the red one is, right? Then you would get a pattern that looks like this, essentially. So there are some features that we can get, but we cannot, by one optimization run, just get this one optimal pattern, right? So then, of course, you can do some kind of cluster analysis, say, right? So you can see how similar are these patterns. Is there any common properties? So here is some projection of the distance between these patterns, so just least square distance between the pattern shapes via multidimensional scaling. I can comment on the method more later, and we can kind of visualize the similarity of the patterns in 2D with this technique, and then we can ask about common properties. So here we can look at the information, and then we can, for example, ask, is this really all local optima, right? Which we claim them to be, and we check gradients and so on. But we can also do perturbations to ask, is there some basins within which these patterns are located in phase space? And that's shown here. So we do some perturbations. This is the solution that corresponds best to data. That's the other ones. And from what we can say is all of these perturbations that we created have information values lower than their optimal values, right? So the optima are the black circles here, and blue circles. And sometimes it seems that we're moving on some kind of manifolds, in the sense that the information values are indistinguishable from the optimal one. But you can look at this more closely here. So here the information is color-coded. And one thing that also we found quite striking is that this solution that comes closest to the measured pattern actually seems to be a really nice hill in that phase space, right? Which means that this is a very localized local minimum, while for some of the other solutions we cannot say that very clearly. So we did still some other types of analysis where we looked at the resource usage of all of these optimal solutions. And resource really means literally number of mRNA and protein that you need to produce these patterns. And there, it turns out that while this solution that comes closest to the Drosophila wild type solution uses, while it produces less positional information, carries less positional information, it also uses less resources. So here we take this resource usage of that solution as 100%, and then we can compare to the other solutions, and you can see that all of them, curiously enough, almost all of them, okay, here we have a lower number, but almost all of them use about 14% more of resources. So one can claim that this Drosophila solution is not unique, but it seems to be among the ones that maximize position information and at the same time try to minimize resource usage, which makes sense from the point of view of the fly, of course, right? So I could show you a couple of other things. We compared, for example, how much information can you generate when you just randomly sample these biophysical parameters and then you compute positional information. It turns out that you hardly ever go above something like 3 bits. So anything above 4 bits must be optimized in a way, right? That's what we can claim for this. But I think in view of time, I will just stop here and summarize. So what I have shown you is that there is this system of gap genes that are interacting, and these encode position information that we can quantify using measures from information theory. there is, which is not said here on the slide, but there is suggestions by analyzing the measured patterns that this system is optimized for encoding position information. Now, we created a biophysically realistic model, I would even say the first biophysically realistic model that can explain these error bars that have been measured in the fly, and also the precision of the resulting precision of the gap change patterns in biophysical terms. We have carried out a large-scale optimization of that model, starting really from first principles, from biophysical principles, and that way we obtained predictions for optimal patterns. The Drosophila solution seems to be among the optimal solutions, but it's not unique. We find also other optima with similar and sometimes even higher information, but typically they use more resources in terms of protein and mRNA numbers. And there remain many open questions from the developmental, biological point of view, systems, and even evolutionary biology point of view, which probably somebody else than me then will have to answer, because that by itself was a large body of work. And now we have created this, as I would say, powerful tool where we can just tune these parameters and we get these patterns and their precision, and we could play all kinds of games, but that's for the future. So let me stop here. Thanks for your attention.
So thinking about how this four-protein system evolved. So previously there was probably one gene and then the second and so on. So probably you can see it in embryological system that first there's one activated, the second, and it should probably correspond to the evolution. And so this is another way of trying to answer these kind of questions. Why this system?
Yeah.
Because first we need to optimize for one gene. Then starting from this we need to optimize for second gene and so on. So maybe this optimization not starting by before ones, but from one at the second, maybe it will lead to other. So
you're having now this picture in mind, exactly, as if this were evolution. Well, I should say here first of all that we are not claiming to evolve the system, because we are not changing base pairs or something. We have a proxy for that, but of course it may be impossible even for evolution to go up that way in general, right, or exactly that way, because there is other constraints on what you can mutate and so on, which we don't take into account. So we are not claiming that this is the same as evolution, but we believe it's a good proxy. Now, the second thing about how many genes were there in the beginning, that we don't know for sure, because we don't have good measurements of species that come before Drosophila, of course, right? So whether there was many or few genes, I'm not sure, because so in the first place you have the input gradient, right? You could imagine that initially there was only gradients, like in bacteria. Bacteria also use internal gradients to specify positions and so on, but of course bacteria are very far away from the fly. So I'm not sure whether I have a good answer to this. I would say it's a good question whether these genes also were put in by evolution sequentially, but I find it very hard to make any claims based on the work that we did.
Sure. I'm saying you don't need to have the answers because it is impossible now. However, it's sufficient to look at embryological development, that this gene are probably activated one by one, and looking at this order, you can suggest how evolutionary it was.
I'm not sure whether you could do any experiment where you mimic evolution this way, but of course what you can do is you can knock these genes out and ask how essential are they for conveying this positional information. And that has been done actually. It's a recent study by, among others, my supervisor Gasper, right, and Marela Petkova, which was published in Cell recently. They have done this, and they came up with a model-free approach where they reconstructed the decoder that optimally reads out these genes, and you can do that in all different settings. So when you have recorded all the mutants also, you can ask how essential are different genes. And it turns out that the system is quite robust, but to attain the precision necessary, you need, as it seems, all four of them. And the precision necessary is this 1%, right?
But just thinking about expression with time of each of them. So there is some plots, and they start probably in different times. And the starting times suggest how the order of interest in the evolution.
It should be understood in a different way, because the optimization algorithm you use, different algorithms would lead to finding different solutions.
Well, even one algorithm leads to finding different solutions.
And what I mean is that maybe some optimization algorithm could be somehow justified on the basis of evolution or the sequence of activation in time or something like this. Because maybe it's like it's minimizing in some direction and only then looking around it. Something like that. But obviously it's
so
difficult to read.
There is one important difference between what I'm doing, and I think that's independent even of the algorithm, right? And what evolution is doing. Evolution only happened, as far as we know, one time on our planet, right? I can repeat this, and I'm repeating this hundreds of times. And still, sometimes I do find these solutions that look like Drosophila, sometimes I don't find them, right? But the point is, evolution, our evolution, depends on history. And that is, the whole history is set at this point, right? Because the algorithm is deterministic, or at least pseudo-stochastic, in the sense that we can always reproduce it, right? And as soon as I'm here, I possibly cannot even create the same evolutionary trajectories that I would have created with a different initial condition, right? So there's no guarantee that I ever find the same initial condition with whatever algorithm I use that evolution has taken. Right? I can possibly find the solutions, but really having the same trajectory, that's much harder, because we know that this phase space has a lot of local minima, right? It's very degenerate. We may get stuck somewhere, and it seems that evolution is happy in finding one of many possible solutions, such as, for example, there's a parallel to deep learning, right? Like, we find one solution, and nobody really understands exactly yet why deep learning works, right? But we're happy by finding one that reproduces our demands, right, on the learning algorithm well enough. And it seems that evolution is also somehow operating in that regime.
It looks like there's another constraint which you don't recognize yet.
That could be, of course, yeah.
Yeah. Yeah, I've got a question regarding your model. You're completely different. So can you get to the slide where you have your time evolution model defined?
The Lagrange Multiplier equation, you mean? Yeah. Maybe this one?
That could be this one. Okay, so basically you did your noise term here is something we call multiplicative noise, because this gamma depends on G. And the huge question is, what kind of noise interpretation you use here?
So it depends on G, but again, I make a linearization assumption, right? So I don't get multiplicative noise in the...
So you just throw it away, you don't deal with it?
The only thing I plug in is the constant average value of G. And I assume, again, there's small fluctuations, right? Such that I can linearize around that. And then everything becomes linear. And I make it some Gaussian assumption as well, right? So
there is a whole range of problems related to this. And basically, two things work interchangeably. The steady state distribution and the noise interpretation. And three things. And the first thing is the fluctuation dissipation relation for this noise. And if you don't know at least two of them, you can say what is the third. And in your case, you don't really know the steady state, I guess, because you want to determine it. And you are not sure what should be the fluctuation dissipation relation, for this. Because I mean, it's
yes.
So,
but then I could answer.
If you do these two things, you would have to determine the third thing, which would be this noise interpretation, and the noise interpretation would introduce another drift terms. So it would change what is your,
Sure,
form.
I know.
But he's mentioned.
I know.
You said encode.
Yeah, but okay, I do this, yes, but at some point I also come up with approximations like the linearization around the means and so on, right?
So say something that can introduce additional forces in this equation, and then should affect the results.
I agree. That this is not the end of the story. It's not so, right? It maybe is not the most detailed model ever. But I need a tractable model. I need one that I can solve in seconds, right? To be able to optimize over these 50 parameters. So I have to make some assumptions. And the one assumption that I make is noise, at least in the optimal solutions, will be small. So the ones that I get on the way, maybe they have the wrong information value because they have the wrong error bar. But I'm interested in the order of magnitude anyhow, right? So for me, what matters is the error bar goes down to 10% for certain sets of parameters. and not to 20. So whether it may be 11, it may be 9, and maybe these things matter. But when everything is small, then again, these approximations should hold, right? Of course, there may be some issues, right? But if it goes wrong, so you're suggesting I may be just missing some essential part of the noise. So noise bars may be locally much higher, for example, because of these effects, right, of multiplicative noise and noise-induced drift.
Deeper physics, related to this, but you can meaningfully talk about it only in the systems where you have, for example, Boltzmann distribution, and it should be there, yeah? Because these drifts are something, in case of this Boltzmann system, yeah? This additional test is something that keeps the equilibrium between different regions, in terms of how particles flow in this case, yeah? But I don't know what would be the analogy for your system, but it has something to do with how this spatial pattern is formed and how it is maintained in this dynamical term. So it is quite important for this, but if your noise is really, this amplitude is very low, it's probably it wouldn't produce anything seriously affecting the system. But still, it could take some.
I mean, the main criticism that you could even bring forward before your comments is that I'm modeling something that is not steady state with a steady state assumption, right?
Yeah.
And that by itself already is an assumption, right? So we assume that it's sufficient to do that because the readout time, so there is a time point where these patterns remain reasonably stable. They never are completely stable, actually, but we make that assumption, right? But then again, everything, all the error bars that you would get in stationary state probably are a good proxy for the ones that you would get over a reasonably long time and so on, right? So if you would have large fluctuations, so here's another counter argument, right? So because your criticism is that I may miss very large, yeah, or suggestions, right? So just you point out that I may miss large fluctuations. Then I would say, okay, that may be the case for the steady state solution, but anyhow, the fly doesn't have time to realize them really. So maybe that would affect one part of the ensemble of embryos that would have to be completely discarded because they are screwed up completely in a way, right? But from what we can see, right, also from the measurements, is that most of them work reasonably well, and they are within that 10% error bar. And okay, it's an a posteriori argument, you may say, but I have created a biophysical model that can explain this based on biophysical parameters, right?
Yeah, but in the grand picture, if you take into account this additional effects, maybe your model changed in a way that the minimum, which is biological minimum for your information at the very end, it would be for some reason more preferable. Yeah, I mean,
yes, sure.
Yeah.
It could impose other constraints, yeah.
Yeah, and it could lead to different results or to some preference for that reason. Yeah,
but I still don't believe that the fact that I'm missing something like noise-induced drift would matter more than creating just crappy patterns, with arbitrary regulatory parameters, right?
Yeah,
maybe we can continue later on.
Because
just
depends on me.
Yeah, I have a few questions I will ask them later on. So
okay.
Maybe let's spend time again.
Thank you.